Hello, and thank you for taking the time to watch my presentation today. I will be covering a recent development in the medical field concerning the treatment of asthma through a review of the journal article from ACS Medicinal Chemistry letters published in 2018, which is titled Discovery of MK-8318, a potent and selective CRTH2 receptor antagonist for the treatment of asthma. In recent years, the pharmaceutical industry has increasingly begun to observe that the chemoattractant receptor homologous molecule expressed on the T helper type 2 receptor gene, also referred to as the CRTH2 receptor, has the capability to be moderated in order to treat allergic reactions as well as asthma flare-ups. Two Merck laboratories identified a molecule called MK-8318 as a potent CRTH2 receptor, which would aid significantly in the treatment of asthma. This is especially a significant development because the current treatment plan for those affected is based on patient compliance and their remembrance to take inhaled steroids multiple times a day. Other oral asthma treatments that are on the market are used secondarily because they have lower clinical efficacy. The use of the CRTH2 molecule as a treatment for asthma can be effective to alleviate both of these problems because it can be taken orally once daily rather than inhaled and is a non-steroidal drug. The majority of this article discusses how the researchers went about creating the final MK-8318 molecule and details the various compounds that they added that worked or did not work to increase the structure-activity relationship called SAR, or pharmacokinetics, also referred to as PK. We'll first discuss the overall reaction synthesis before starting back at the beginning of the process that researchers took to determine the final compound. So the reaction synthesis to create the final compound is overall on the screen right now. They began with a compound of ethyl 2-oxocyclopentane carboxylate, which was reacted with the base 4-fluoroaniline in the presence of zinc to create an amino ester. This amino ester then underwent hydrolysis to create an acid, and this acid was converted to a ketone by a treatment with Eaton's reagent. The ketone was then treated with Bach protection to keep it protected and from reacting when the race-big mixture of the molecule was resolved to be exclusively cis-race-big, using chiral supercritical fluid chromatography, also referred to as SFC, in order to create an enantiomerically pure isomer. Cyclopropylamine was then introduced to reduce amination and was acylated, resulting in an amide. The Bach protection on the southern portion of the molecule was then removed using trifluoroacetic acid, as you can see here, and they then coupled this intermediate molecule with 4-trifluoromethoxybenzoyl chloride, which then underwent hydrolysis to give the final product 15C, as you see here. Overall, the researchers stated that this process is highly efficient and allowed them to produce analogs easily. Beginning back at the very beginning, conformational analysis of this first compound initially showed researchers that compound number 3 here could be a very promising potent CRTH2 ligand, especially as a binding assay, working well to improve interaction between the receptor and the molecule. However, upon functional camp assay, the compound appeared to actually be a functional partial agonist. Hopeful that they could improve reactivity, researchers compared it to indomethacin, a known CRTH2 agonist in a homolog molecule, because the two had very similar functional groups. Upon comparison with the model of indomethacin with a similar structure, it was postulated that the aniline phenyl group in compound 3 might be overlapping with the methoxybenzene in indomethacin, which occupies the agonist pocket in that molecule. They also discovered that in the modeling study, that there are two very important hydrogen bond interactions, which are known to be crucial to structure-activity relationship activity. Knowing this, a small ethyl group was introduced on compound 4 to minimize the interaction in this northern portion of the molecule, as you can see right here. And they noticed that this worked very effectively and also opened up several positions to improve structure-activity relationships between molecule 4 by identifying the essential pharmacophores in the northern, middle, and southern portions of the molecule. Starting with the northern section, they noticed that cyclobutyl and cyclopropyl derivatives 5D and 5E, as you can see here and here, were the best accepted lipophilic alkyl groups. Polar substitutions in this northern area of the molecule weren't super well accepted because they occupied the lipophilic region of the molecule. They decided to use the cyclopropyl 5E because it had better binding activity and also the potential for better structure reactivity. They also added an aminocarboxylic acid side chain that they found to be important to increase and decrease reactivity. They found that molecules 6 and 9, these two were the best reacted ones, but they determined that the 2 carbon chain was more effective, compound 9 here, because it significantly improved reactivity, and the farther from the amide carbonyl the carbamic acid was, the less reactive it became. So structure 9 was the one that they continued their further modifications with. Moving on to the center and southern sections, these two, they sort of popped back and forth between. They would optimize one section and start to optimize the southern section of the molecule before realizing that it was actually better to change up the northern section, and then they would go back to the southern section to see if they could continue to improve this reactivity and binding agents. Initially, they determined that cyclopropyl and cyclohexyl fused rings had better binding activity, but the structure activity relationship was super narrow. So they attempted to add some small substitutions in order to increase the pharmacokinetic properties, but this only resulted in less activity from the molecule as a whole. So they decided to fix the cyclobutyl ring to this core portion of the molecule, and further develop the structure activity relationships of the southern portion of the molecule to see if further optimization could be achieved despite limitations in this center portion. So they went down to the southern section, and initially they saw some amide substitutions in the southern portion to be potentially promising. Aryl amides specifically were tolerated pretty well by the molecule and initially appeared to increase potent binding and reactivity in the molecule. The southern aryl amides also appeared to work well with the acid side chain that we discussed earlier on the northern portion of the molecule. But when they moved to rat PK studies, all of the rats had super poor exposure, especially when the carbamate substitution 12E and 12F on this previous slide, this one and this one, they did not react well. So these findings caused the researchers to re-examine the cyclopentyl fused core in the center of the molecule to see if improved PK could be achieved. So back to the center, they determined that amide functionality in the southern portion might be necessary. And so they continued testing in rats. They did in vitro metabolite testing, which is testing to determine what reaction pathway the molecule is taking. And they determined that it was glucocoordination, also known as detoxification in the body by making the molecule water soluble, which is why the PK range was so low. And while reviewing this PK data, rediscovered compound 5D, which is on slide 5, which they hoped would allow them to find compounds which would increase the plasma exposure and just increase reactivity in the body as much as possible. So they decided to move forward with this as opposed to something else because they believed it would help increase the reactivity. Because of this, they began to modify the center phenyl ring, which they hadn't quite done before, and they found that adding a fluorine group, specifically 15C, they found as super effective. They found all of them to be pretty effective, but 15C stood out as being the most effective. It retained super strong biological activity when put in the body and done in tests on rats, as well as giving improved exposure when tested in clinical settings. They attempted some further optimizations on 15C to see if they could continue to increase the reactivity, but nothing worked. And so they ended up just moving forward with 15C to see if they could get any satisfactory results in their discussion and continued research. It demonstrated really good pharmacokinetics, such as bioavailability, and this molecule specifically had an adequate half-life in order to allow for only once daily dosing in humans, which is lower than the standards for many other asthma medications right now. 15C also showed clean profiles in ancillary studies and met all of their potency assay target profiles. So because it passed all of their tests with flying colors, they began to perform preclinical testing in vivo. And here we are. The CRTH2 molecule 15C, they tested in rhesus monkeys, sheep, dogs, and rats. I couldn't find anything on the monkeys or dogs in the article, but they discussed their findings in the sheep and the rat trials specifically, and they tested throughout the article, as I've been mentioning, in rats to determine PK levels. In the sheep model, they did this trial intravenously, and they found that it blocked late airway responsiveness as well as airway hyperresponsiveness, which are two very indicative reactions with asthma. And this was administered at a dose of 1 milligram per kilogram of body weight. And in ovalbumin sensitized brown Norway rats, which is ovalbumin sensitization is the reaction with a form of egg white to rats to cause sort of artificial airway responsiveness. So that way they can perform drug trials on these animals and get accurate sort of asthmatic responses without trialing on humans yet. And they found that in brown Norway rats dosed orally at 310 and 30 mpk, it provided very good efficiency in improving airway function, which is huge. And so I wasn't able to find anything online on if they've continued preclinical trials or if it's moved on to human clinical trials. But either way, why did I focus on asthma? In 2019, the World Health Organization estimated that 262 million people worldwide are affected by asthma. I am specifically affected by this research and also by asthma. I have moderate asthma, and I have many friends who also struggle with asthma. One of whom is allergic to her inhaler, but doesn't have another option to use when she has an asthma attack. Her option is either have her asthma attack get worse or use her inhaler and have an adverse reaction. So taking an oral drug like this is much more likely to have a better reaction for her than the one that she's currently stuck with. This article stood out to me when I was doing my initial research on an article to do because the overview and purpose of this article, they discuss this medication as a promising future treatment for mild asthma as a non-steroidal and orally dosed alternative to the standard practice of prescribing inhaled steroids that need to be taken twice or more daily. And a struggle faced by doctors and patients alike is the ability to consistently and effectively take their medication. I personally have really struggled with remembering to take my medication. And it's hard to keep the bulky inhalers on you at all times if you're not necessarily around them when it's time to take your dosage. So having something like this that's able to be taken once daily orally is much better for stuff like traveling because it's easier to transport and just easier to have around. And just you need to remember to take it fewer times a day. So specifically, this drug could be used by me or various friends of mine who have mild asthma several years down the line, assuming that it does become in the market as a potential treatment for asthma. I'm hopeful that this drug could continue to be trialed. Yeah, it would be fascinating and really exciting to see it as a treatment for asthma in the future very soon. Yeah, thank you. Like and subscribe.